Vaccines are one of the most significant advancements in modern medicine, playing a crucial role in preventing infectious diseases and saving countless lives. By introducing a weakened or inactive form of a pathogen into the body, vaccines stimulate the immune system to produce antibodies without causing the disease itself. This process prepares the immune system to recognize and fight off the actual pathogen if encountered in the future. The concept of vaccination dates back centuries, with the practice of variolation in ancient China and India to prevent smallpox. The first successful vaccine was developed by Edward Jenner in 1796, using cowpox to protect against smallpox. This breakthrough laid the foundation for the field of immunization and led to the eventual eradication of smallpox worldwide. Vaccines have evolved over time, with advancements in technology allowing for the development of vaccines against a wide range of diseases, from polio to measles to influenza. The success of vaccines lies in their ability to create immunity within populations, leading to the concept of herd immunity. When a large percentage of a community is vaccinated, it helps protect those who cannot be vaccinated due to age or medical reasons. As we delve deeper into the world of vaccines, it is essential to understand the science behind how they work and the critical role they play in safeguarding public health. The history of vaccines is a fascinating journey that spans centuries and has had a profound impact on public health. The concept of immunization dates back to ancient civilizations, where practices such as variolation were used to prevent smallpox. Variolation involved exposing individuals to small amounts of infectious material to induce immunity. This method, although risky, laid the groundwork for the development of modern vaccines. The breakthrough in vaccination came in the late 18th century when Edward Jenner successfully created the first vaccine against smallpox. Jenner observed that milkmaids who had been infected with cowpox, a less severe disease, seemed to be immune to smallpox. He tested his hypothesis by inoculating a young boy with cowpox and later exposing him to smallpox, proving the effectiveness of the vaccine. This pivotal moment marked the beginning of systematic vaccination efforts. Smallpox vaccination campaigns were launched globally, leading to the eventual eradication of the disease in 1980. The success of the smallpox vaccine paved the way for the development of vaccines against other infectious diseases. In the 19th and 20th centuries, vaccines were developed for diseases such as rabies, typhoid, cholera, and polio. The discovery of new technologies, such as cell culture and genetic engineering, revolutionized vaccine development, making vaccines safer and more effective. The establishment of organizations like the World Health Organization (WHO) and the Centers for Disease Control and Prevention (CDC) further accelerated global vaccination efforts. Vaccines have played a crucial role in preventing epidemics and saving millions of lives. For example, the introduction of the measles vaccine led to a significant decline in measles-related deaths worldwide. Despite the remarkable progress in vaccine development, challenges remain, including vaccine hesitancy and access issues. Continued research and public education are essential to ensure the success of vaccination programs and protect communities from infectious diseases. Principles of immunization revolve around the fundamental concepts of how vaccines work to stimulate the immune system and provide protection against infectious diseases. The primary principle of immunization is to introduce a weakened or inactive form of a pathogen, known as an antigen, into the body. This antigen mimics the infectious agent, but does not cause the disease itself. When the immune system detects the antigen, it mounts a response by producing antibodies and activating immune cells to target and eliminate the perceived threat. This process creates immunological memory, allowing the immune system to recognize and respond rapidly if exposed to the actual pathogen in the future. Vaccines can contain different types of antigens, such as live attenuated viruses, inactivated viruses or bacteria, subunit vaccines, and toxoid vaccines. Each type of vaccine triggers a specific immune response tailored to the characteristics of the pathogen. Live attenuated vaccines contain weakened forms of the virus or bacteria that can replicate in the body but do not cause disease. Inactivated vaccines consist of killed pathogens that cannot replicate but still stimulate an immune response. Subunit vaccines contain only specific antigens or proteins from the pathogen, focusing the immune response on key targets. Toxoid vaccines target bacterial toxins by using inactivated toxins to induce immunity against the harmful effects of the toxin. Another essential principle of immunization is the concept of vaccine schedules and booster doses. Vaccines are often administered in multiple doses to ensure optimal protection and long-lasting immunity. Booster doses help reinforce the immune response and extend the duration of protection. Vaccination schedules are carefully designed to maximize the effectiveness of vaccines and provide immunity at critical stages of life. Immunization programs follow evidence-based recommendations from health authorities to determine the most effective vaccine schedules for different populations. Understanding the principles of immunization is crucial for appreciating the significance of vaccines in preventing infectious diseases and safeguarding public health. Vaccines come in various types, each designed to target specific pathogens and elicit an immune response tailored to the characteristics of the disease. Live attenuated vaccines contain weakened forms of the virus or bacteria that can replicate in the body, stimulating a robust immune response. Examples of live attenuated vaccines include the measles, mumps, and rubella MMR vaccine and the oral polio vaccine. These vaccines often provide long-lasting immunity with a single dose, but may not be suitable for individuals with weakened immune systems. Inactivated vaccines consist of pathogens that have been killed or inactivated, such as the polio vaccine and the hepatitis A vaccine. While inactivated vaccines are safe for most people, they may require booster doses to maintain immunity. Subunit vaccines contain only specific antigens or proteins from the pathogen, focusing the immune response on key targets. The human papillomavirus HPV vaccine and the acellular pertussis vaccine are examples of subunit vaccines. These vaccines are highly purified and have a reduced risk of side effects compared to whole-cell vaccines. Toxoid vaccines target bacterial toxins by using inactivated toxins to induce immunity against the harmful effects of the toxin. The tetanus and diphtheria vaccines are toxoid vaccines that protect against the toxins produced by the bacteria. These vaccines are crucial for preventing severe diseases caused by bacterial toxins. Recombinant vaccines are a newer type of vaccine that uses genetic engineering to produce antigens that mimic those of the pathogen. The hepatitis B vaccine is a recombinant vaccine that contains a protein derived from the hepatitis B virus. Recombinant vaccines are highly specific and can be produced more efficiently than traditional vaccines. Vector vaccines use a harmless virus or bacterium as a vector to deliver antigens into the body and stimulate an immune response. The COVID-19 vaccines developed by Pfizer-BioNTech and Moderna are examples of mRNA vaccines that use lipid nanoparticles as vectors to deliver genetic material encoding the spike protein of the SARS-CoV-2 virus. These vaccines have shown high efficacy in preventing COVID-19 infection. Understanding the different types of vaccines is essential for developing effective immunization strategies and combating infectious diseases. The development of vaccines is a complex and rigorous process that involves multiple stages to ensure safety, efficacy, and regulatory approval. The journey from initial discovery to widespread vaccination can take years and requires collaboration between scientists, researchers, regulatory agencies, and pharmaceutical companies. The vaccine development process typically begins with the identification of a target pathogen, followed by research to understand the pathogen's characteristics and how it interacts with the immune system. Scientists then explore various vaccine platforms and technologies to design a vaccine that can effectively stimulate an immune response. Preclinical studies are conducted to evaluate the vaccine's safety and efficacy in animal models before moving on to clinical trials in humans. Clinical trials are conducted in multiple phases to assess the vaccine's safety, immunogenicity, and effectiveness. Phase 1 trials involve a small number of healthy volunteers to evaluate the vaccine's safety and dosage. Phase 2 trials expand the study to a larger group of participants to further assess safety and immune response. Phase 3 trials are large-scale studies that involve thousands of volunteers to evaluate the vaccine's efficacy in preventing disease. Regulatory agencies review the data from clinical trials to determine if the vaccine meets safety and efficacy standards for approval. Once approved, manufacturing processes are scaled up to produce the vaccine on a mass scale. Quality control measures are implemented to ensure the vaccine's consistency and purity. Post-marketing surveillance continues after the vaccine is released to monitor for any adverse events and assess long-term effectiveness. The COVID-19 pandemic has accelerated vaccine development timelines, with multiple vaccines authorized for emergency use within a year of the pandemic's onset. The rapid development of COVID-19 vaccines highlights the importance of collaboration, innovation, and global cooperation in responding to public health emergencies. Vaccine development is a dynamic and evolving field that continues to push the boundaries of science and technology to protect populations from infectious diseases. Herd immunity, also known as community immunity, is a critical concept in public health that occurs when a significant portion of a population becomes immune to a disease, either through vaccination or prior infection, providing indirect protection to those who are not immune. This phenomenon helps prevent the spread of infectious diseases and protects vulnerable individuals who may not be able to receive vaccines due to medical reasons. The threshold for achieving herd immunity varies depending on the contagiousness of the disease. For highly contagious diseases like measles, a vaccination coverage of around 95% is typically needed to establish herd immunity and prevent outbreaks. Herd immunity not only protects individuals, but also helps control the spread of diseases within communities. During the COVID-19 pandemic, achieving herd immunity through vaccination has been a primary goal to control the transmission of the virus and reduce the burden on healthcare systems. However, reaching herd immunity for COVID-19 has been challenging due to factors such as vaccine hesitancy, unequal access to vaccines, and the emergence of new variants. Understanding the principles of herd immunity is essential for designing effective vaccination strategies and promoting public health. By working together to increase vaccination rates and protect vulnerable populations, we can strengthen herd immunity and create safer communities for everyone. Vaccine safety and efficacy are paramount considerations in the development and deployment of vaccines to protect public health. Before a vaccine is approved for use, it undergoes rigorous testing to ensure that it is safe and effective. Vaccine safety is monitored at every stage of development, from preclinical studies to post-marketing surveillance. Adverse events following vaccination are carefully monitored and investigated to assess the vaccine's safety profile. Vaccines are held to high safety standards, and regulatory agencies require extensive data on safety and efficacy before granting approval. Despite the meticulous safety protocols in place, vaccines, like any medical intervention, can have side effects. Most vaccine side effects are mild and temporary, such as soreness at the injection site or low-grade fever. Serious side effects are rare, but are thoroughly investigated to determine if they are directly linked to the vaccine. Vaccine efficacy refers to the ability of a vaccine to prevent disease under ideal conditions. Clinical trials are conducted to evaluate a vaccine's efficacy by comparing the incidence of disease in vaccinated individuals versus unvaccinated individuals. The effectiveness of a vaccine can vary depending on factors such as the individual's immune response, the circulating strains of the virus, and the timing of vaccination. For example, the flu vaccine's efficacy can vary each year based on how well it matches the circulating flu strains. Despite these variations, vaccines remain one of the most effective tools for preventing infectious diseases and reducing the burden of illness. Vaccines have been instrumental in controlling and eradicating diseases such as smallpox, polio, and measles. The development of COVID-19 vaccines in record time has demonstrated the power of scientific innovation and collaboration in responding to global health crises. Vaccines not only protect individuals, but also contribute to community immunity, reducing the overall spread of disease. By upholding high safety standards and continuously monitoring vaccine efficacy, we can ensure that vaccines remain a cornerstone of public health interventions. Vaccines have revolutionized public health by preventing infectious diseases and saving millions of lives. From the first smallpox vaccine to the rapid development of COVID-19 vaccines, the history of immunization is a testament to human ingenuity and scientific progress. Understanding the principles of vaccination, the different types of vaccines, and the importance of herd immunity is crucial for protecting communities. Vaccine safety and efficacy are paramount, with rigorous testing ensuring that vaccines meet high standards. As we continue to navigate global health challenges, vaccines remain a cornerstone of disease prevention and control, highlighting the power of vaccination in safeguarding public health.